easy. As Kristen said, we are Team Bass, and we are so excited that all of you could make it to our presentation today. So our thesis is the appropriateness of largemouth bass as a model species for the detection of endocrine disruption. Presenting today are Shivani Patel, Renuka Tripu, Laura Wilson, and myself, Shafali Shah. The rest of the team members are Christine Kim, Taylor Crow, Winston Liu, and Angela Leska. So our story begins on the southern branch of the Potomac River. The Potomac River flows into the Chesapeake Bay and is home to a large variety of organisms, including the large and smallmouth bass. In 2002, researchers started noticing that large numbers of smallmouth bass were dying. When they opened up these fish for histological examination, they realized that the male fish were exhibiting both male and female sexual characteristics, which is the condition that is referred to as intersex. The largemouth bass in the area also had a high prevalence of intersex, so they were otherwise healthy. We decided to use the largemouth bass in our experiment because even though they have one of the highest rates of intersex in the United States, they haven't been well studied in a laboratory setting. So largemouth bass usually completely sexually differentiate into either males or females. So the fact that they're intersex is highly unusual and is representative of a global pollution problem. Therefore, we wanted to use largemouth bass as a model species to study endocrine disruption in a laboratory setting. So because intersex is such a big problem, it's really important to really nail down the causes of why it's happening before we can start to manage the problem. So I touched on it a little bit earlier, but what exactly is intersex? So over here we have a slice of a male fish gonad. So the pink tissue is normal male tissue. However, you guys can see those big blobs in the corner over there that are purple. Those are actually eggs. So these males have eggs in their testes, which is something we call testicular oocytes. So this is one biomarker of intersex. Another biomarker of intersex is the presence of vitellogenin. Vitellogenin is a female egg protein that females produce in order to provide nourishment for their young. So both male and females have estrogen receptors on their liver. When estrogen binds to these receptors, vitellogenin is produced and ultimately eggs are produced. So because females naturally endogenously produce their own estrogens, they are going to also produce vitellogenin. However, because males don't produce vitellogenin normally, the fact that they are means that they're being exposed to some sort of endocrine marker outside in the water. So you guys are probably thinking to yourself, what exactly is causing this intersex? Why is it happening? What's going on? So the short answer is that everything poops. Chickens poop, cows poop, we as humans poop, and this poop is a natural source of steroid hormones like testosterone and estrogen. This waste can find its way into the water system, and from there it can influence the endocrine system of other organisms. At this point, these compounds become endocrine disrupting compounds, or EDCs for short. So we have a couple examples of some EDCs up here. The first is 17-beta-estradiol, or E2, which is a potent estrogen found in poultry litter. Over time, it will convert into a less potent form called estrone, and this will be important for the later part of our presentation. The other EDC we have is ethinyl estradiol, or EE2, and this is a synthetic estrogen that's found in birth control. So how exactly are these EDCs getting into the water system? So intersex prevalence is usually higher downstream from point sources of pollution like wastewater treatment plants versus upstream. So this suggests that industrial sources may be one cause of intersex. So here's a map of Maryland and the Eastern Shore. So all the red and yellow dots, those represent residential areas and also industrial sources. And if you look on the Eastern Shore of Maryland, you'll see that there aren't really these industrial sources. However, this is where we're finding the most intersex. So we were thinking to ourselves, what could be causing the intersex here? So research has also shown that intersex is correlated with seasonal changes, which suggests that agriculture could play an important role in causing it. In fact, the Eastern Shore of Maryland produces 1.6 billion pounds of poultry litter every single year. So that's a lot of poultry litter. And because it's so easy and accessible, farmers extensively use it as fertilizer on their land. So when it rains, this poultry litter could wash into the bay, and as I mentioned earlier, it contains natural estrogens, specifically 17-beta estradiol, and we hypothesized that this is what was causing the intersex that they were seeing. Poultry litter has also been shown to induce and increase the telogenin levels in male fathead minnows. So the fathead minnow is a common laboratory fish. It grows really fast. It is visually sexually dimorphic, so you can easily distinguish males from females just by looking at them. And it grows really well in the laboratory. So we wanted to make sure that we included this species in our experiment. So the three big research questions that we had were, one, can exposure to poultry litter induce endocrine disruption in largemouth bass, specifically in the form of testicular oocyte development or increase the telogenin levels? Two, was there an age of range or window sensitivities that these fish were most susceptible to developing endocrine disruption? And finally, three, how does the endocrine sensitivity of the largemouth bass compare to that of the fathead minnow? So as I mentioned earlier, it's been shown that you can increase the telogen levels in the fathead minnow in the laboratory, but we're really seeing this intersect in the largemouth bass in nature. So we wanted to see if largemouth bass could also be used as a model species to study intersex, because that is what is representative of what is actually happening in nature. I will now pass it on to Shivani to talk about methodology.
So how exactly did we answer those three research questions? Well, over the past 2 years, we conducted a total of 4 laboratory exposures that consisted of different species of fish, as well as different ages of fish, and as well as different treatments of fish. And by exposure, I just mean that we took fish, put them in large tanks of water that had something in them for a period of time, and then we studied what happened to them afterward. As you can see in our timeline, we conducted 3 of our exposures in 2013, and these 3 exposures had a similar experimental design. Exposure 4 occurred a year later, and it had a slightly different design, and I'm now going to each of these in detail. So exposure 1 occurred in June of 2013. We used 12-month-old largemouth bass, and as you can see, we put about 50 in each treatment. So, as Shefali mentioned earlier, we wanted to investigate poultry litter as a contributor of endocrine disrupting compounds that was causing intersex in these fish. So, our main experimental treatment was poultry litter. And how we prepared that mixture was we essentially took a bunch of chicken poop and grinded it up, as you can see Winston doing here. And then we took about 1 kilogram of that mixture, put it in 2,000 liters of water with the resulting 0.5 gram per liter concentration. We also had a negative control of diluent dechlorinated city water, and this was just used as a comparison to the other 2 treatments. Finally, we have a positive control of a known active estrogen, 17-beta estradiol, and at a known target concentration of 50 nanograms per liter. And this is just a setup in our laboratory of those 3 treatments I just mentioned. You can see the negative control, positive control, and poultry litter. And what's important about these treatments is that they were also the same for exposures 2 and 3. And another important thing to note is that these were static exposures, meaning we put in the poultry litter mixture, we put in the estradiol, and we let it sit for the 14-day period. The concentrations were not replenished. And this is significant because 17-beta estradiol, as Shefali mentioned earlier, converts into estrone. So the concentration of estradiol on day 14 was much less than what was present on day 0. And exposure 2 was conducted in September of 2013. We used 3-month-old largemouth bass. Again, we have the same 3 treatments at the same concentrations. The only difference here was the age of the bass used. Next, we had exposure 3 in November of 2013. We actually exposed 3 different groups of fish simultaneously. We had 6-month-old and 18-month-old largemouth bass. And then after our first 2 exposures, we decided to add fathead minnows to the mix. So what's important about fathead minnows is that they've already been proven to be an excellent model species for inducing endocrine disruption in the laboratory. So we included them as a comparison to our largemouth bass. Finally, we have exposure 4 in November of 2013. We used 3-month-old largemouth bass and 9-month-old mature male fathead minnows. So you can see this one looks a little different. We did not use any poultry litter in this exposure. Instead, we used 3 different concentrations of 17-beta estradiol. We had a low concentration of 2 nanograms per liter, a medium of 9 nanograms per liter, and a high of 30 nanograms per liter. And in terms of concentrations found in nature, 2 is not on pretty high, but has been found to occur, and 30 is pretty unheard of. But the purpose of our laboratory exposure was not to replicate environmental conditions. Instead, we wanted to induce that effect of endocrine disruption and study it. And here's just a diagram of what exposure 4 looked like. So if you recall earlier, I mentioned that exposures 1 through 3 were static exposures, meaning that we put in the culture litter, we put in the estradiol, and it sat there. Exposure 4 was not static. Instead, we used a flow-through system so that the 3 concentrations were actually maintained throughout the entire 14-day period. I will now hand it off to Renee to talk further about our methods and data analysis and collection.
Thank you, Shivani. I'm going to talk about what we actually obtained from these exposures. So like Shivani mentioned, we exposed our fish for 14 days. Periodically through this exposure period, we collected samples from each of the treatments so we could perform chemistry analysis and make sure we were maintaining an adequate exposure environment. So what we did is we collected 2 samples from each treatment on days 0, 1, 3, 5, 7, 10, and 14 of the exposure. We filtered them down to 0.7 microns, and then we put them through solid phase extraction in order to concentrate and purify the samples for the 2 tests that we were going to perform. One of these tests was a test for estrogenicity. Estrogenicity is how active and potent the estrogens in our treatments were. So for this, we used a bioluminescent yeast estrogen screening assay, also known as the BLIES. This is essentially a yeast strain with human estrogen receptors that will identify those extraneous estrogens and will light up in response to them. So again, this was used so we can determine exactly how potent our treatments were. We also wanted to detect the different types of estrogens that were in the treatments. So as Shivani and Shefali mentioned earlier, 17-beta estradiol will convert into estrone. So we wanted to quantify the amounts of both of these throughout our exposures. For this, we used tandem mass spectrometry. So this will separate the different types of estrogen by mass so we can see the patterns of them throughout the 14 days. Okay, so after this 14-day exposure period was over, we did 2 things. We either sacrificed a subset of the fish, or we grew them out for a couple months. For the fish that were immediately sacrificed, we wanted to collect vitellogenin egg protein, as we talked about earlier, and this would have been only available immediately after the fish were exposed. So what we did is we anesthetized the subset of fish, and we obtained blood from their caudal vein. We then centrifuged this blood and obtained the blood plasma. With this plasma, we performed the enzyme-linked immunosorbent assay, also known as the ELISA. This assay works in kind of an antibody-antigen relationship. So what this means is that it will there will be antibodies that will kind of attach to that vitellogenin with a fluorescent label. So it will light up in correlation to exactly how much vitellogenin was present in the blood plasma. With the fish that we grew out, we wanted to collect their gonads. So we were mostly concerned with testicular oocytes in male testes, but we did collect both species or both genders of gonads. So as you see in these pictures, we have the male testes. Oops, sorry. Wrong button. Up here, this is the male testes, and then we have the female ovaries. So we collected both, and then with the testes, we put them 3 cross-sections of them on slides, and then we had 3 of our team members go through a double-blind analysis, which means they did not know the treatment or the age of any of the fish they were looking at, and they analyzed them and looked through for these testicular oocytes. So for exposure 4, one of the differences in addition to the treatments was how we collected our data. So actually we collected and sacrificed fish periodically throughout the exposures and collected blood plasma and vitellogenin that way. So we sacrificed fish on days 4, 7, 10, and 14 throughout the exposure period and collected vitellogenin, in addition to performing the typical water chemistry analysis as we did previously. Now I'm going to pass it on to Laura, who will talk about all of our results.
Thank you, Rita. So I wanted to begin by introducing the results of the BLIES analysis for estrogenicity for exposures 1 through 3. As you recall, these exposures were static, meaning that the estradiol positive control in the experimental poultry litter were added on the first day of the exposure and allowed to sit for the entirety of 14 days. Since exposures 1 through 3 have the same treatments, their BLIS results were all very similar. And here we have the results from exposure 2 as representative for all 3 exposures. On the horizontal axis of our graph, we have the number of days into the exposure that the sample was taken. And on the vertical axis, we have the estradiol equivalence, or EEQ, which is how estrogenic the sample is compared to a similar concentration of estradiol. Our negative control is shown in the blue, the poultry litter in the yellow, and the estradiol positive control in red. We notice in both the positive control and the poultry litter that the estrogenicity is generally decreasing over the course of the exposure, and that the estradiol degrades from sitting in the exposure tanks. However, the diluent water control has a slightly increasing trend from day 7 to day 14. This could be due to the crowding of the fish within their tanks, which exposes them to the estrogens that they naturally excrete. Also, we notice that the positive control is higher than the poultry litter over the entire exposure. And this is favorable since we wanted to compare our experimental poultry litter treatment to one that we know is very estrogenically active. So now that we have shown that our positive control in our poultry litter samples were estrogenic, we wanted to look at the results of the tandem mass spec to determine what compounds are present that have caused this estrogenicity. Here we have the results from the mass spectroscopy done on the positive control samples collected during exposure 3. On the horizontal axis, again, we have the days into the exposure that the sample was collected, and on the vertical axis, we have the concentration of each estrogenic component. The dark blue shows the 17-beta estradiol, that potent estrogen that was mentioned before, and the red shows the concentration of estrone and the less potent estrogen. And the result of the BLIS estrogenicity is overlaid in green. Notice the estrogenicity measured in BLIS follows the same trend as the concentration of estradiol, which is what we would expect, since the estradiol is the most potent estrogen and is going to contribute the most to the estrogenicity. We also see that as the estradiol concentration decreases, the estrone concentration increases, which is showing that conversion from estradiol to estrone that was mentioned before. We then compare those results to that of the experimental poultry litter treatment. The results showed that the poultry litter did have some estradiol which degraded over the exposure. However, the poultry litter had a much higher concentration of estrone, which increased from day 1 to day 4. This additionally suggests that there are other estrogenic components present within the poultry litter that are converting to the estrone. So how did these estrogens affect our fish? These are the vitellogenin results from exposures 1 through 3. Recall that vitellogenin is the egg yolk protein that we're using as a biomarker for the physiological response to estrogen. So on the horizontal axis, we have it broken up by the fish species, age, and exposure number. And on the vertical axis, we have the average concentration of BTG within the fish exposed. And I wanted to note that the BTG is on a log scale. The blue represents the fish within the negative control, the red the poultry litter, and the yellow the positive control of estrogen. We noticed that there was a lot of variability within this data as shown from the tails on the bar graph, even within this log scale. We also noticed that there was no observable trend between age and treatment type for the largemouth bass. However, the fat minnows, which we ran concurrently with the largemouth bass in exposure 3, shows a very clear correlation between BTG production and treatment type. But remember from our BLIS results, these fish were only exposed to a potent estrogen for a few days at the beginning of the exposure. So this led us to think that maybe the largemouth bass just need more time with a more potent estrogen to promote this BTG production, which led us to our 4th exposure. So these are again the estrogenicity results from the BLIS for exposure 4. The horizontal axis shows the days into the exposure, the vertical axis again shows the estradiol equivalents. And from the bottom, we have the control treatment in blue, the low treatment in red, the medium in yellow, and the high in the brown. And as you can see, we did a very good job at maintaining relatively stable estrogenicity for each of the treatments that were distinct from one another. We then looked at the concentrations of estradiol and estrone using the tandem mass spectroscopy. The horizontal axis is lit up by the 4 exposure treatments, the control, low, medium, and high. And the vertical axis shows the average concentration of each estrogen in the exposure. The blue shows the 17-beta estradiol, the most active estrogen. The red shows the estrone, and the green shows the estrogenicity measured from glides. Again, we notice that the estrogenicity follows the concentration of estradiol, and also we notice that with more estradiol in the sample, more conversion to estrone happens, causing more concentrations of estrone with estradiol. The BCG results are broken into exposure, treatment, and duration. And we noticed with the largemouth bats that all the values were universally low, and there was no observable trend between the treatment type or duration. However, with the fathead minnow, there does show a trend based on the dose of estradiol. For the negative control, low, and medium treatment, they were not able to promote significant ECG expression, but the high treatment was able to for all durations. And I will now pass to Shivani to discuss the histological results.
Okay, so I'm going to quickly talk about the last part of our results. So in addition to collecting water samples and blood samples, we also collected that gonad tissue we were talking about to look for testicular oocyte development, specifically in males that were grown out after exposure. The reason for this is because TOs do not develop immediately, they need some time to develop. So up here we actually have some pictures of our own fish. Even though the fish did not respond well to the intelligent production in our exposure, we still found testicular oocyte, which was interesting. And like I said, you can see our pictures here. What was interesting to see is that they varied in both severity. So some only had one, whereas others had 6 or 7 in a bunch. And here's just a table summarizing the testicular oocytes we found. So of 96 males that were grown out and sacrificed across our first 3 exposures, we had 9 developed testicular oocytes. And it was interesting to see that these occurrences of fish did not follow any sort of pattern. For example, we had 3 control fish in exposure 1 develop testicular oocytes, which is bizarre because our control didn't have anything in it. And this random distribution maybe suggests that some sort of outside factor may have predisposed these fish to developing testicular oocytes. Maybe it was the hatchery conditions in which they were grown in. And maybe that 10% prevalence rate was just a normal background occurrence. I'll now hand it off to Radhika to finish up our presentation.
So we were able to achieve a lot in our 3 years on this project. Not only did we build our lab from scratch, but we also exposed over 800 fish to the treatments we talked about earlier. We also optimized all of our assays for our analysis specific to our experiment, which involved a lot of work. One of our main concerns was whether or not we could provide an adequate exposure environment for our fish, meaning whether or not we were actually exposing them to estrogens. Based on the results of our chemical analysis, as well as the concentration-dependent response in the fathead minnows, we kind of concluded that yes, we did provide an adequate exposure environment for our fish, although the largemouth bass did not respond in the same way. But this could be due to a lot of reasons. Maybe the largemouth bass were just not amenable to our own lab conditions. Maybe we didn't provide enough exposure time, enough exposure treatments. Maybe we should have varied the ages a bit more. Maybe due to the hatchery conditions they were born in and the dense cultures, they just didn't respond as we expected. The bottom line is, we're seeing a lot of this intersex in largemouth bass in nature, but we just weren't able to replicate it in a lab setting. But this is also really exciting because it provides a lot of ground for future research to be done in this similar topic because it is very, very important. But none of this would be possible without the help of a lot of people. I want to point out just a couple. So our discussants that are here with us today and that read through our thesis, Ms. Nevenka Bravkovska, who was our librarian, Sea Grant, and the Gemstone staff. But most importantly, the person that has been most instrumental to our project is our mentor, Dr. Yankos. He was absolutely essential in our project from day 1 till the very end, staying up with us late throughout nights and days during our exposures, making presentations, writing our documents, listening to us sing Disney songs as we cleaned the lab. He was there for all of it, so we really cannot thank you enough. But now we will take any questions that you guys might have. Thank you. We can do the introductions and we'll do the relevant questions.
My name is Steve Gurley. I'm a aquatic toxicologist with the University of Maryland Wine Research and Education Center in the heartland of poultry litter.
I'm Fred Pinkney with the U.S. Fish and Wildlife Service in Annapolis, Maryland, doing environmental reporting.
I'm in Orlando and with the Department of Animal and Avian Sciences, and much of my research actually overlaps a lot of what they've done here. Although recently we're working more with compounds, other compounds that are more controllable besides
Scott Lynn, I work for the U.S. EPA and the Office of Science Coordination and Policy.
Dr. McGee, you want to ask the first question?
Can you speak a little bit more about background levels of intelligence or maybe this is the early science? It seems like, as you mentioned, that you actually found that in the controls you actually saw this particular early science. So is there any literature that would suggest what sort of is a natural level of it within marshmallow packs?
So the question was if we can speak a little bit more about background levels of ETG and oocytes. I'm just going to cover oocytes because I'm more familiar with that. Dr. Vicki Blazer, who we hoped would be here today but couldn't make it, has published, or actually she's in the process of publishing research that suggests there's between a 5 to 10% background incidence of testicular oocytes. Our specific prevalence was 9.4%, so it falls in that range, so that's what led us to that conclusion.
And as mentioned with the BTG before that, it is a female egg-yolk protein precursor, so it shouldn't be shown in males. And when we were working on optimizing our protocols for measuring BTG, we ran into a lot of issues with how to set a detection limit and how to quantify the levels that we're finding. And I think it's more so an anomaly of assay that we're finding this BTG in our controls than it is something that is naturally occurring in the wild.
Thank you.
Yes. If you were to run a follow-up study to this, what changes would you make? Or what direction would you go in if you were to follow this up with another study?
So you can go next slide. So the question was, are there any like future directions that we would take this in? And you can see some of them in there, but I think probably the biggest thing we'd like to do is, we're looking for this age window of sensitivity. And initially, we really thought the age that's going to happen is just when that sexual differentiation occurs. So really, really young fish, just after they've been hatched. And due to a variety of reasons, we weren't able to do that. So that's probably the first thing we try to do. Try to look at that age that we think actually should be the one that's the most causable. So that's probably the first thing you can see all sorts of other things that we're going to do as well. Thank you for that question.
I guess we sort of had a similar issue when we got smallmouth bass from the hatchery, and we had this beautiful experiment, and we did something analogous to what you did, and we caged them and put them in rivers, and we found that our pre-exposure fish had more testicular oocytes in the river. So I guess how do you deal with, this is sort of more of a personal question, how do you deal with the frustration of for why and finding X?
All right, so I guess that was an interesting question. So he was asking, how do you deal with frustrations in research in general? Is that kind of where the question is? I actually think frustration is one of the most beautiful things about research. Because it's when your hypotheses don't work, it's when everything's going wrong that you are starting to see new things that are exciting. That's when you're going to make that breakthrough. If everything just goes swimmingly according to what you think is going to happen, I don't think you're breaking through anything, you're not discovering anything new. I think it's those times when, for example, you're discovering oocytes where you're not supposed to discover oocytes. Maybe there's some factor that you're not considering. Maybe there's something really exciting that you can actually focus on next time to really understand that phenomenon. So I think that's how I would answer it.
So I really have to keep that in mind.
Yeah, I'm frustrated.
I love your attitude. So one question is about these testicular oocytes. So they may be there's something that's unnatural, but yet there may be some background element in this fish, in other fish. What's the harm? I know that it's unnatural, perhaps, right? Like they're supposed to have separate sexes, you're supposed to have ovaries, ovarian tissue, and testes, and testicular tissue, but you got a few oocytes inside testes. What's the harm? Is there harm?
So the question was, what is the potential harm of testicular oocytes? And actually, the whole reason that we started this project is because there was a large amount of fish kills throughout the Chesapeake Bay, and they were actually discovering that a lot of these fish, once they cut them open, have these testicular oocytes. And there's recently been a paper suggesting that there might be a connection with immunity suppression and these testicular oocytes. So there is a potential connection which needs to be investigated further, but we didn't go that far in our experiment.
So a lot of people have asked about testicular oocytes. I'll switch gears and I'll ask a question about the Bly's assay. My understanding of the Bly's assay is that it has human estrogen receptor alpha that is activated when there is an indicator that shows up. There's a lot of conservation invertebrates in terms of steroids and that hormonal pathway. My question, though, really focuses on the fact that it's estrogen receptor alpha specific. Fish are, I think, the only vertebrates that actually have 3 estrogen receptors as opposed to 2. So I just wanted to see if you guys wanted to comment on this and talk about why the Bly's assay was chosen and why you think it's actually relevant to what you're doing.
All right, so I can take that one. That's great, fantastic question. We love answering tough questions, right? So if I'm understanding you correctly, you're talking about the fact that Bly's assay is based on a yeast strain that is not matches to that, where that responds to the alpha receptor, as opposed to fish would actually have more than just the alpha receptor. So can the Bly's assay actually be looking at estrogenicity levels in the water? And that's your question. I think it's a great question. I think I'll start by saying the reason we chose the Bly's assay is because we had seen some literature published about using the Bly's assay as a measure of estrogenicity. Dr. Wanowitz, who also I think works very closely with Dr. Blazer, has been using the Bly's assay and has been using it very well and showing these levels. And so that's the reason why we approached that. I think it's a great point to bring up that maybe, down the road, that's something you may want to consider and look at different receptors and different types of estrogens. But for us, at least at this stage, this is just what we're doing. So I don't know if that sufficiently answers your question. But, okay.
If you knew then what you know now, would you do it? From doing an engineering project.
So the question was if we knew then what we know now, would we do it again? And I think one of our biggest problems at the beginning was we aimed really high. So as you can see on this slide, there's the non-lethal testing with the laparoscopy, and we hope to do a field study. So those were 2 things that were included in our original proposal and just were ideas we were really excited about when this project first came about. And I think maybe if we had started off with a much smaller aim and then only hoped to move to that if we got that far, that probably would have been a more realistic approach and might have allowed us to get into a little more depth with some of these methods and fish agents and such.
I just want to say that this was an amazing amount of work. And I read through this and I was like, oh my gosh. So I just want to acknowledge you for your work. It is a little frustrating sometimes you don't get what you're thinking going in, but you got a great attitude that excited to get you.
Maybe here in the back, be commended for the great amount of work that Nancy did. Welcome. Do you have a few minutes for questions from the audience? I'd like to open it up at this point. Here, and then here, and then Joe.
So what did the literature say to you about the conversion of estradiol to estrone in the field? Is it bacterial driven? Is it a UV driven conversion? And would it alter how you set up your design in the future?
So the question was, how does the conversion from estradiol to estrone happen in the field? And there's another part.
And how would it change how you would set up your design of your experiment?
Would it change the design of the experiment? So the conversion from estradiol to estrone happens from reaching a microbial environment that is going to cleave away some of the side groups and the rings of the estrogen, which eventually causes it to form the estrone and eventually degrade. So when we introduced it to our exposures with the fish naturally have microbes popping all over them that want to eat that, cleave off those side groups and eat them, and which causes the estrogens to activate. So our experimental design kind of mimicked the natural setting in that way.
My question is, how did you come to determine that 14 days was an appropriate time for exposure, and how confident were you that that was enough time to see results?
Okay. Great question, Eric, and thank you for asking. Sorry, it's audience, right? Okay. So the question was, why 14? Why the magical number 14? I think the short version of that, the answer to that question is the feasibility. Obviously, the best way to do something like this would be to do really long exposures, mimicking seasons, and all sorts of good things like that. But if you're in a lab, in the animal science building, trying to do this over a 2-year project, 14 days seemed like the most reasonable time frame for us. And yeah, in the literature, there's some literature that has done similar types of things as well. But I think feasibility was the biggest limiting factor there.
I was wondering how certain it was that intersex is causing this diminishing population. For example, could it be that intersex is actually improving the survivability, so non-intersex fish are dying off, so there's simply more intersex fish in relation?
So is the question that the fish that are actually dying were not intersex, and so the ones that the research were looking at did have intersex and were surviving better?
Well, yeah, I'm asking if there's any survival bias, that there's just now a higher population of intersex fish.
So originally, sorry, go ahead.
Yeah, go ahead.
So originally, the researchers examined fish that were already dead. So those are the ones that were having intersex. And so I think Taylor touched on this a bit earlier, but what really people think is happening is that endocrine exposure to estrogens, it's causing lethality in fish species, but it's also causing intersex. So they both are markers of sort of a bigger problem of endocrine disruption. So the testicular oocytes, nothing has suggested that that's actually what's causing the deaths in these fishes, but it's likely that they're both caused by the same thing. And does that answer your question?
Yeah.
Okay, thank you.